Hello everyone. Today we're going to talk about statistical significance. It is defined as the probability that test results have a real difference and it's not due to chance.
What are the factors that may affect statistical significance? First is the size of a sample. A larger sample is more suitable to represent a normal population and achieve more statistical power. Second, the scatter of the group samples. This means how the results are distributed. The first graph illustrates how two groups should be further apart from each other to show a difference, and each of them contains similar results. In the second graph, the two groups share similar results and each result is distributed broadly, making it harder to demonstrate an actual significance.
What is it based on? It is important to remember that in research, it is impossible to analyze the whole population. Consequently, we obtain our data from a sample. It is possible to do this based on the normal population distribution or bell-shaped distribution. The midline represents the mode, median, and mean. If we assume two standard deviations from each side, then it will be included 95% of the population. This is the base to understand the confidence interval. The confidence interval is a range of values within which the true mean of the population is expected to be based on a specific probability. Thanks to this, we are able to resample the results from our data to the general population. To keep in mind, if the interval confidence contains 0 or 1 in the case of relative risks or odds ratio, then the null hypothesis cannot be rejected.
In a normal distribution based on a z-test, it is important to remember that results can be one-tailed, meaning they go one way, either right or left. For example, comparing one medication that will reduce an outcome with another medication. If the results are two-tailed, it means they go in two ways. For example, results on a test that either can have a high or low score. In this example, the results of the investigation is a right one-tailed test. In the normal distribution of the population, a z-value is obtained from our own data through calculations. Based on this value and analyzing through a specific table of z-values, a p-value can be obtained. The p-value is defined as the probability of obtaining a sample more extreme than the ones observed in your data, assuming a null hypothesis distribution. A p-value that is lower than 0.05 favors the rejection of the null hypothesis and accepts the alternative hypothesis. This means there's little likelihood that the observed difference between the sample mean and the hypothesis mean is due to chance. A p-value that is higher than 0.05 favors accepting the null hypothesis and rejecting the alternative hypothesis.
But how do we evaluate the statistical significance? There are specific tests that compare means useful for this purpose. Some of them compare quantitative means, such as t-test, that compares two groups only, or ANOVA, that compares more than two groups. And there is also tests that compare qualitative category means, such as chi-square or z-test. In summary, statistical significance is the probability that test results have a real difference and it's not due to chance. It can be affected by scatter or size population. It is based on confidence interval and p-value. And how do we measure it? With various tests such as t-test, ANOVA, chi-square, and z-test.